Mouse embryos were bred on the International Space Station. In an experiment conducted on the International Space Station, mouse embryos were grown. The embryos developed normally in microgravity. No significant changes in DNA were observed in additional tests in laboratories on Earth. The study, designed by molecular biologist Teruhiko Wakayama of Yamanashi University with support from the Japan Aerospace Agency, JAXA, tested whether mammalian embryos can develop normally in a microgravity environment. For this purpose, frozen mouse embryos were flown to the International Space Station. After thawing them, the astronauts grew the embryos for 4 days under the watchful eye of scientists on Earth. They were then preserved and sent back to the planet for analysis. Experiments on the International Space Station suggest that early embryo development is not affected by conditions of low gravity and increased radiation. The results and description of the research were published in the journal, iScience. Wakayama and his colleagues carried out the first stages of the experiment in their laboratory on Earth, taking early-stage embryos from pregnant mice and freezing them. The frozen embryos were sent to the ISS in August 2021. They were stored in special equipment designed by Wakayama's team so that astronauts on the station could easily thaw the embryos and culture them. The astronauts then chemically preserved the embryos and sent them to Earth on the return ship. Scientists found that embryos grown in microgravity conditions usually developed into blastocysts, i.e., cells from which the fetus and placenta develop. They added that the research has clearly shown that gravity has no significant effect on early embryo development. They also stated that after analyzing the blastocysts sent back to their laboratories on Earth, they did not observe any significant changes in the DNA. This is the first ever study to show that mammals can thrive in space, scientists from Yamanashi University and the Riken Institute said in a joint statement. This is the world's first experiment in which early-stage mammalian embryos were grown in microgravity conditions on the ISS, they added. The embryos were only cultured for 4 days because that's only how long they can survive outside the uterus, Wakayama admitted. His team studied embryos sent from space to Earth to see if their development was affected by exposure to the higher radiation and low gravity of space. The embryos showed no signs of DNA damage from radiation exposure, the researchers said, perhaps because they were only in space for a short time. They showed normal structural development, including differentiation into 2 groups of cells forming the basis of the fetus and the placenta. This was an important discovery because it was previously thought that microgravity could affect the ability of embryos to separate into these 2 different cell types. Many frozen mouse embryos flew into space. A control group remained in laboratories on Earth. The survival rates of the group of embryos on the ISS were lower than those of the control group. But those embryos that succeeded developed normally. Embryos cultured in microgravity developed into blastocysts with normal cell number, normal internal cell mass, trophectoderm, and gene expression profile similar to those cultured on Earth, the researchers wrote. It is unclear whether being in space would disrupt later stages of embryo development. Previous studies in which NASA sent pregnant rodents into space for 9 to 11 days during the second half of pregnancy showed that they gave birth to offspring of typical body weight. The young later developed normally. Those studies and our results show that perhaps mammal reproduction in space is possible, admitted Wakayama. However, other research indicates that gravity loading is necessary for proper musculoskeletal development. This type of research could be important for future space exploration missions. I show, as the authors of the works claim, that getting pregnant in space can be safe for people. It is possible to get pregnant during a future space trip, for example to Mars. The journey there will take over 6 months. We are conducting research to make sure that we will be able to have children in such conditions, said Teruhiko Wakayama. The study of mammalian reproduction in space is essential to the launch of the space age, making it necessary to study and understand the impact of the space environment before the ISS ceases to operate, the scientists said.